Today we'll be looking at the physiological framework of cell deterioration as animals mature and age. Imagine a world where we can slow the aging process and enhance the vitality of our pets in their golden years. While the fountain of youth remains a dream, recent advancements in understanding the biology of aging reveal promising pathways. Today we'll explore the physiological factors behind cellular deterioration and identify key mechanisms that drive aging, highlighting potential interventions to promote healthier, longer lives for our beloved animals. As animals age, their body stem cells, which are essential for tissue regeneration, begin to decline in function and lose their ability to communicate effectively, leading to the visible signs of aging. Concurrently, their mitochondria, the energy producing structures in their cells, start to malfunction, generating harmful free radicals that contribute to oxidative stress, further damaging their cells and DNA. Additionally, the protective caps on their chromosomes, known as telomeres, shorten with each cell division. When they become too short, they compromise chromosomal stability, resulting in DNA damage and accelerating the aging process. Together, these factors significantly impact the overall health and longevity of our animals.
Stem cells can be directed to differentiate into particular human usable cells. The body can regenerate those cells and restore tissues that have been damaged or impacted by disease through the process of synthesis. But as we age, adult stem cells lose their functional potential and become less healthy. Some examples of stem cells include self renewing cells called somatic stem cells that are found in human tissues and organs during development. Neural stem cells are cells that are mostly present in the central nervous system's neural tissues. Mesenchymal stem cells, with the capacity to differentiate into several subtypes of tissues, include osteoblasts, cartilage cells, myocytes, and adipocytes. With great capacity for regeneration, adult skeletal muscle stem cells can arise following an injury or as consequence of memory B cells and naive T cell depletion. Hematopoietic stem cells are those that produce blood through the process of hematopoiesis. They are located in the red bone marrow, which is the marrow cavity of most bones.
Hemopoietic stem cells also produce immune cells for organisms. Alterations may come from external factors and reduce their capacity to renew. Stem cells can be altered or reversed by systemic or circulatory components obtained from youthful plasma via anatomical joinings or parabiosis. Parabiosis allows for modulating the molecular structure of the tissue environment rather than inducing cells alone. Other treatments include cellular reprogramming to create induced pluripotent stem cells, or iPSCs, which are a type of stem cell produced from specific adult somatic cells. This process allows the biological clock to reset by eliminating certain traits from disease cells associated with aging.
Let's now discuss the physiological effects of oxidative stress and mitochondrial dysfunction. Cells' mitochondria start to malfunction with age due to the oxidative damage to cellular components. This happens when mutations accumulate in a mitochondrial DNA and over time impair the mitochondria's ability to produce energy effectively. Mitochondrial dysfunction leads to increased production of high reactive molecules called free radicals or reactive oxygen species, ROS. This feedback loop continuously generates more ROS, which creates a vicious cycle of cell dysfunction. ROS can cause widespread damage to cells' proteins, lipids, and DNA, which disrupts normal cellular functions. The excessive ROS generated overwhelms the cell's antioxidant defenses, leading to oxidative stress. Now let's discuss some potential interventions that are seen in long-lived animal species that tend to have higher basic activities of antioxidant enzymes. One potential intervention is enhancing cellular antioxidant systems to better neutralize ROS, which can be done by supplementation of antioxidants like vitamin E, vitamin C, and CoQ10. Another potential intervention would be to restrict calories and dietary alterations. Caloric restrictions in animals show reduced mitochondrial ROS production and lower oxidative stress to mitochondrial DNA.
I want to talk about a significant proponent of aging, which is genomic instability. As we age, our cells pick up the genetic equivalent of scars from internal and external threats. This damage presents itself in a multitude of ways, from mutations to chromosomal alterations to telomere degradation. While our bodies have mechanisms to repair this damage, these mechanisms decline as we age. One of the key players in this process is the telomere, which functions as a protective cap at the end of our chromosomes. Each time a cell divides, the telomeres shorten, and when they become critically short, cells can no longer divide and enter what is known as cellular senescence. This plays a significant role in the aging process. While the enzyme telomerase can preserve telomeres, it is absent from most somatic cells. Reducing the sheltering complex, which guards telomeres from being recognized as double-strand DNA breaks and thus halts chromosome fusions, illustrates how essential it is to preserve telomeres and chromosome fitness. The systemic viral transductions of telomerase into mice has been shown to delay normal physiologically associated aging and thus offers an increase in lifespan. These findings raise hopes for potential new therapeutics to prolong a healthy lifespan by manipulating telomere maintenance pathways.
As animals get older, their cells start to degenerate, and they will soon become susceptible to any disease that their body seems to contract. When that happens, their cells tend to age as well, and that means their bodily functions will soon stop working how they should. You all, as future scientists, should continue your research and gain more knowledge on more ways we can possibly prevent older animals from dying of degenerative cells. We love our animals, and seeing that as they age, they become more of a target to diseases. We want to make sure that our pets are active and up to date on all vaccines, just like us. We want to live a long, healthy life, so why can't we find more ways to do the same for animals?